Okay, let's see. Can you guys see this? Can you see the slideshow? Are we good? It should work, hopefully. I don't know, but it should work. Okay, Ray can see it. Can everyone else see this? Yeah? Okay. Okay, so we'll get started then, because I'm gonna assume no one else is coming. If they are, they can join in the middle. Still works. Okay, so we're gonna do Unit 3, which is basically the SL section of genetics. I'll also eventually do Unit 10 later on, and then that will cover the HL area. So the first thing we need to know in genetics is what a gene is. So a gene is a heritable factor that consists of a length of DNA and influences a specific characteristic. So any section, a gene is basically a section of our DNA, and it is something that is targeting to one specific characteristic. A gene is also something that occupies one specific position on one type of any type of chromosome. The various specific forms of a gene are called alleles, and alleles differ from each other by one or a few bases only. So alleles are basically different types of the same gene. So if a gene is coding for eye color, for example, alleles are the different types that can result from that code. So it can be blue, green, brown, hazel, etc., etc. And the differences between each of these alleles in terms of their DNA composition will only be differing by one or two bases. It won't be more than that. Oh, by the way, if you guys have any questions while I'm explaining something, just put them in the stage chat because then it'll be easier for me to look at while I'm still speaking. Okay. So, about mutations. So, new alleles are formed from other alleles by gene mutation. Mutations are completely random changes. There's no mechanism for a particular mutation to be carried out. So there's no like specific procedure that's followed when a particular mutation is being carried out. The most significant type of mutation is base substitution. This happens when one base in the sequence of a gene is replaced by a different base. So if, for example, the sequence of bases is supposed to be AGCT, and instead of AGCT it becomes AGCA, that's basically what base substitution means. So when this happens, obviously, since you're changing over one of the bases, it changes what the allele is, and that's when mutations come about. So almost all mutations are either neutral or harmful. There are very few mutations that are actually positive, and we don't learn about them. And mutations can also be lethal, so they will cause the death of the cell in which the mutation occurs. And this happens because that base sequence of that DNA is just not viable for living, which means that it will end in the death of that cell. So the main case study that we have for mutations is sickle cell anemia. So in sickle cell anemia, we have basically it's a condition where the red blood cells of that person, instead of being a normal shape of a button, it becomes sickled, or um, basically becomes very thin, and therefore can't carry oxygen, which means that it causes an overall reduction in their oxygen perfusion and obviously other issues as well. So I'm not going to go into, I'm not going to explain the basics of how it works, but we all should know by now how the codons work. So instead of the glutamate codon, we get the valine codon, which makes the red blood cells sickled. So that it's basically an issue of base substitution. So this is another, it's a type of mutation that causes an issue in people with sickle cell anemia. And so here's how it works. So when it's in a normal gene, in the DNA, the CTC gets paired up with the GAG. Um, I mean, that's what the normal DNA is. And then when it's being transcribed, the CTC will become GAG, and then in the mRNA, and then when it gets paired up with the tRNA, it gets paired up with a CUC, which is glutamic acid or glutamate, and that's what codes for normal hemoglobin. But when it's mutated, the DNA sequence will have a CAC pair with a CAC against a GTG instead of a CTC against a GAG, which means that the A and T are swapped, which is a substituted base. So when it goes through transcription and becomes mRNA, it becomes GUG instead of the GAG, which means that the codon on the tRNA becomes CAC, which will code for valine, which changes the amino acid sequence or the protein sequence from glutamine or glutamic acid to valine. So that's what causes sickle cell anemia. Yeah, so as you can see, instead of glutamic acid, it becomes valine. And those are two completely different chemicals. You don't need to know the structure of these chemicals, by the way, but I just put them there so you have a visual of how it works. Their base is the same, but um, there's other groups. If any of you take chemistry, there's other groups involved rather than just the main strand, and those are changed. So when that's changed, it changes the um, shape of the red blood cells. Is there any questions about sickle cell anemia before I go on? No? I'm going to assume that means no. Okay, so the next one is genome. So the genome is the whole of the genetic information of any organism. It's basically the entire DNA sequence, essentially. It's every bit of the genetic information that any organism will hold. So in humans, the genome consists of the 46 molecules that form the chromosomes in the nucleus, plus the DNA molecule in the mitochondrion. It's not just the DNA that exists in our chromosomes in our cells, but it's not just from the nucleus, it's also from the mitochondria, because our mitochondria, as we know, carry DNA as well. In plant species, we know that both the mitochondria and the chloroplasts have DNA in them, which means that the genome for plant species would be the chromosomes in the nucleus plus the DNA molecules in both the mitochondrion or the mitochondria and the chloroplast. So the human Genome Project was when in from 1990 to 2003, and they some the scientists coded the entire base sequence of human genes, and it gave us data that can be analyzed for patterns. So it basically is something that's used for analysis of patterns and how gene sequence and base sequencing works. And another discovery was that most of the genome is not transcribed. So through the Human Genome Project, we found out that a lot of our genome is just not transcribed. There are elements that affect gene expression, as well as highly repetitive sequences that are called satellite DNA, which don't actually they're not actually expressed, but they are what help mRNA, tRNA to be they basically help in the gene expression but are not expressed themselves. So they help in locating certain genes, if you will. Okay, 3.2 chromosomes. So in a prokaryote, there's one chromosome consisting of a circular DNA molecule. So in any prokaryote, it's a circular DNA molecule. It's not just a string of DNA. So the DNA is naked, which means that it is not associated with smaller proteins and it's not in a nucleus-like structure or in any other structure. Some prokaryotes will also have plasmids, which are much smaller, but they're extra loops of DNA, which will be also again in a circular shape. Them being in circular shape basically means that they're not in conventional chromosome shape as we know in humans. So they're basically in a string that connects into a circle. So the difference between eukaryotic and prokaryotic chromosomes, this is kind of important. It can come up as a question, just as a question to answer. So eukaryotic chromosomes and prokaryotic chromosomes. The first difference is eukaryotic chromosomes have linear DNA molecules. Prokaryotic chromosomes have circular DNA molecules, which means that they don't have a particular place of start and end. It's basically it keeps going in a circle. But in eukaryotic chromosomes, it is a linear DNA molecule. Also, in eukaryotic chromosomes, they are associated with histone proteins, which means that they're not naked. They're associated with histone proteins, which basically help in the coiling. And in prokaryotic chromosomes, they're obviously, like I said, naked. They don't have any associated proteins. They're just in their circular molecule. In eukaryotic chromosomes, or the eukaryotic genome doesn't have any plasmids, but in prokaryotes, there's often plasmids, which basically are part of the chromosomes. In eukaryotic chromosomes or eukaryotic organisms, there's usually two or more different chromosomes, but in prokaryotic organisms, there's only one chromosome. Not associated by proteins. Yes, I mean histones. Sorry, I answered that here. Okay, so chromatids. Now, each chromosome in prophase and metaphase of mitosis. I'm not going to explain what mitosis is and how it works, but you should know that already. So in the prophase and metaphase of mitosis, each chromosome will consist of two structures known as the sister chromatids. So I don't know if you guys can see my cursor. I don't know. Let me see. No? Wait. Yeah, okay. You should be able to see my chromosome. So my cursor. Now, that's one sister chromatid and that's another sister chromatid. So if you split it down the middle, there's one here and one over here. These each contain a DNA molecule whose base sequences are identical. So this part of the chromosome is identical to this part of the chromosome, and that's why they're known as sister chromatids. They are held together by a centromere, which is the point where the sister chromatids are joined, and which is also the point that gets split apart during mitosis in anaphase. Okay. Next, we're going to talk about homologous chromosomes. Now, it's important to remember that homologous chromosomes and sister chromatids are two different things. So, homologous chromosomes carry the same sequence of genes, but not the same alleles. So, let's go back. In a chromatid or a chromosome, the gene sequence and the alleles are the exact same on each of the sister chromatids. So if you split it down the middle, this is exactly identical to this with the gene sequence and with the alleles that are being represented. However, in homologous chromosomes, the gene sequence will be the exact same, but the alleles does not have to be the same. So, for example, in this pair of homologous chromosomes, these two pink ones, pink and purple. So these two pink ones have the same exact gene sequence and the same exact alleles. So do these two purples with each other. However, this pink and this purple one will have the same exact gene sequence, but not the same exact alleles presented. So in humans, for example, there's 23 different chromosome types, but 2 chromosomes for each type, which is why we have pairs of chromosomes. So we have 23 of this type of chromosome, or we have one of this type of chromosome, one of another type of chromosomes, but then for each of that type, there will be 2, which will create the pair, which is why we say we have 46 chromosomes, or 23 pairs. So the pairs will carry the same gene, but not the same allele, like I already explained. Okay, now we're going to talk about haploid and diploid or diploid, however you like to say it, nuclei. These are often represented by N and 2N, which will be useful in a lot of situations. So a diploid nucleus or a diploid nucleus is when the nucleus contains pairs of homologous chromosomes. And almost all body cells have diploid nuclei or diploid nuclei, and they will obviously it'll be homologous chromosomes. They're in pairs. They're what help. That's why they're in most body cells, because that's how it causes mitosis instead of meiosis. But in haploid nuclei, there's only one chromosome of each type, and it's only in gametes, such as the sperm or the egg cells of humans. They're haploid. So they will undergo meiosis, and they basically don't have diploid nuclei. They have one chromosome of each type, and that's it. The next is chromosome numbers. So it's fairly simple. It's self-explanatory. The number of chromosomes, which is usually diploid, is a characteristic feature of members of a species. Different species will have different number of chromosomes. Humans have 46, chimpanzee has 48, dog has 79, 78, etc., etc. These are just some examples. So one of the things that we can draw from this is that because humans and chimpanzees have such a similar number of chromosomes, a lot of our characteristics are similar, which is why we are often considered very closely associated with their species, because our number of chromosomes and our type of chromosomes are very similar. So, which is also why you can see that we don't really associate ourselves as being similar to dogs in characteristic, like in terms of physical characteristics, because the way they're coded for and the way we're coded for is fairly different. Like they have 78 chromosomes, we only have 46, which means that there's a fair bit of difference between our gene sequencing and theirs. However, us and chimpanzees, like I said, fairly similar. Okay, sex determination. Now, this is very important when we're talking about, especially when we're talking about sex-linked diseases, but in general, sex is determined by the sex chromosomes, which in humans is the 23rd pair. Autosomes are chromosomes that do not determine sex. So we have autosomes and sex cells or sex chromosomes, which are the two different types, which is why we have autosomal diseases and sex linked diseases, and we'll go more into that later on. But in humans, obviously, the 23rd pair of chromosomes, either the XX or the XY chromosome, determines the sex of an individual. So in females, it's XX, and in males, it's XY. Okay, now we're going to talk about karyotypes and karyograms. The karyotype is the number and type of chromosomes present in a cell or organism. So it is basically, it's self-explanatory. It's the number and the type of chromosomes that are present in any organism cells. A karyogram is a photograph or diagram in which the karyotypes are shown. So the chromosomes of any organism are shown in homologous pairs of decreasing length in a karyogram. Karyograms are basically very useful in determining either the sex of an individual or diagnosing conditions that are due to chromosome abnormalities, for example, Down syndrome or trisomy 21, which we will talk about. So, the next topic is meiosis. Before I start this, do we have any questions? No? Nothing? Okay. Okay, I'll keep going. Cool. So, in meiosis, there is, the first thing is fertilization. So, a male and a female gamete fuse together to produce a zygote. Meiosis, however, is the process that halves the chromosome number and generates haploid gametes. So, because in fertilization a male and a female gamete fuse together instead of and bring their set of chromosomes or half of their chromosomes, meiosis is a process that allows the chromosome number to be halved and generate haploid gametes, which therefore then fuse together to create a diploid zygote. In meiosis, a diploid nucleus will divide twice to produce 4 haploid nuclei. So in meiosis, there's basically double division. So there's meiosis 1 and meiosis 2. So in meiosis 1, obviously we have prophase, metaphase, anaphase, and then telophase 1. So in prophase 1, it works essentially how normal prophase works. The chromosomes will condense, the nuclear envelope will start breaking down. But this is important. This is where crossing over will occur. We will talk about crossing over later on. But this is basically the step where crossing over could possibly occur. So in this, obviously, again, like I said, chromosomes condense, and then in metaphase 1, those condensed chromosomes and the pairs of homologous chromosomes move to the equator of the cell. Now, when they do this, obviously, while they do this, the centromeres or the centrosomes will move to the opposite sides of the cell during prophase 1, and then in metaphase 1, they will release spindle fibers or microtubules that will go and attach to the pairs of homologous chromosomes. In anaphase 1, these homologous chromosome pairs will move to the opposite poles of the cell and will get ripped apart, basically, but the sister chromatids stay together. So we will, at the end of meiosis 1, essentially have 2 cells without homologous chromosomes, but the sister chromatids still intact. Again, the chromosomes will gather at the poles of the cells, the cytoplasm will divide. However, unlike mitosis, the nuclear envelope will not form again. That will happen after meiosis 2 ends. However, there is cytokinesis, the 2 cells will come apart. Meiosis 2, in prophase 2 of meiosis 2, a new spindle forms around the chromosomes. Like I said, the nuclear envelope doesn't really form. So it will essentially have the chromosomes be there, new spindle forms around the chromosomes, or the centromeres go, centromeres? No, centrosomes go to the opposite poles of the cell. In metaphase 2, again, the chromosomes will all line up at the equator. And then in anaphase, the centromeres will divide and the sister chromatids will move to the opposite poles of the cell, which basically means that now they're no longer the they're not a chromosome anymore. It's hard to explain. They're not exactly the same chromosome anymore since their sister chromatids have been separated. And then in telophase 2 and cytokinesis again, the nuclear envelope forms around each set of chromosomes, and the chromosomes are only the single sister chromatid. And then the cytoplasm will divide, and then now we have 4 since these 1 become 2, and then the 2 divide to become 2 more, which results in 4 haploid cells. Do we understand meiosis? Before we talk about crossing over and genetic variation as a result. Are we good? I'm going to assume the silence means yes and keep going. Okay. So we have genetic variation. There's 2 main ways that we talk about in terms of genetic variation. There's 2 main ways that it can happen. The first one is the random orientation of pairs of homologous chromosomes in metaphase 1. So if we go back, in metaphase 1, there's homologous chromosomes and they are paired up together. However, which homologous chromosome goes to which side is undetermined. So there can be different combinations of chromosomes produced, and therefore different combinations of alleles produced based on where the homologous chromosomes go. And the second thing is the number of possible combinations of chromosomes produced by random orientation. The number of possible combinations of chromosomes produced by random orientation is 2n in humans, where n is 23. Which means that should say 2 to the power n. Hold on, I'm going to fix that. That's on me. No. I'm going to fix that because then I'm going to forget that I didn't fix it. Yeah. So, the number of possible combinations of chromosomes produced by random orientation is 2 to the power n in humans, where n is 23, which means there's over 8 million combinations of chromosomes per parent. Which means that there's just general genetic variation there because, for example, let me go back to the last one. If we talk about, if we go here, so let's say, as we can see here, the 2 blue ones and 1 red one are on this side, and the 2 reds and 1 blue is on this side. However, it could also be that this was the red one and this was the blue one, but those different combinations will result in different parents, which automatically creates a lot of genetic variation. The other way we have genetic variation is during crossing over in prophase 1. So in prophase 1, we have the pairs of homologous chromosomes present in the cell. This is where crossing over happens, where essentially the same section of the chromosome of the closest most sister chromatids cross over and get exchanged between the 2 chromosomes. So, like you can see here, that the same section of blue and purple are swapped over in the pair of homologous chromosomes, which means that the alleles are different now. Does everyone understand genetic variation? Do I need to over-explain this? Yes? I'm going to take no message as yes. Okay. The next is non-disjunction. So non-disjunction is what causes certain genetic problems, such as uh, trisomy 21. So this is when chromosomes that should separate and move to opposite poles during meiosis do not, and instead move to the same pole. This can happen at two times at both the anaphases, during anaphase 1 or anaphase 2, and this will basically result in gametes being produced with more or less chromosomes. So in anaphase 1, there is a possibility of the homologous chromosome pair not separating properly, and both of them ending up in anaphase in one of the cells, and one of the cells only having not having that chromosome at all. So in these situations, the chromosome that does not have the one of the chromosomes ends up dying, and the one that has extra will usually cause some kind of issue in the future, such as possibly trisomy 21. It can also happen in anaphase 2, where instead of the sister chromatid separating properly, the centromere does not come apart, and therefore the entire chromosome gets dragged to one side, which means that if when this gets split into four, this one will survive. This one, however, will die since it has one less chromosome, and these two obviously are healthy. So this basically is where trisomy 21 happens, where in the 21st chromosome there are three chromatids instead of two. Okay, I explained it way too early. I forgot that the slide was right there. Okay, so trisomy 21 or Down syndrome is when gametes with one chromosome too few quickly die, but then gametes with one chromosome too many, like we talked about here, sometimes survive. And when they fertilize, a zygote will be produced with three chromosomes of one type instead of two. And this is where trisomy happens. So trisomy 21, as this is a karyogram, we can see that in the 21st position, there's three chromosomes instead of the two that there should be. So, for example, children are sometimes born with three chromosomes of type 21, which causes Down syndrome. This can be due to non-disjunction during the formation of either the sperm or the egg. It does not have to be one specific one. Now we're going to talk about chromosome testing. There is two main types of chromosome testing that we need to know about. And these are chromosome testing relating to children. So there is the amniocentesis or centesis. I've never figured out how to pronounce it, but as long as you know how to spell it, it does not matter. Amniocentesis or chorionic villus sampling, so we need to know both of them. Amniocentesis or centesis is when a sample of the amniotic fluid in the amniotic sac when a mother is pregnant is removed from is taken from around the fetus, and then that sample is used to do the genetic coding or the sequencing, sorry, to see the chromosomes. And then the other type of chromosome testing is the chorionic villus sampling, where cells will be removed from fetal tissues in the placenta, called the chorionic villi, and this usually happens. It usually happens also while the mother is pregnant. However, this does have a slightly higher risk of miscarriage than amniocentesis, and that's obviously because you're dealing with the placenta, which is where the baby is attached, and any issues with the placenta can cause major risks for the fetus rather than amniocentesis, which will involve less of an issue. Um, but the difference is only 1%. Both of them are fairly um, like safe in terms of causing a risk of miscarriage. Both methods have a very small risk of infections in maternal or fetal tissues, so it's fairly safe to do it. Okay, the next thing, oh, we're taking a break. Right. So instead of making it 5 minutes so that we finish faster because there's a lot to go through, I'm going to make it a 2 minute break. If there's any questions, let me know. I'm going to drink some water. Yeah. Okay, I'm just going to continue because I don't think there's any questions and I've now hydrated. So, we're going to talk about inheritance and how that works. Not the inheritance of money, the inheritance of genes and hereditary characteristics. So, the first person we need to know is this man on the right, whose name is Gregor Mendel. And he is often regarded as the father of genetics because a lot of his research is what helps us in figuring out inheritance. Nowadays, his crossing of the varieties of pea plants, as you should know, the pea plant experiments um, deduce the principles on which inheritance is based. Um, yeah. So the first thing we need to know regarding inheritance and the entire process of it is dominant and recessive alleles. So there's two types of alleles. There's dominant alleles and recessive alleles. Dominant alleles are the ones that mask the effects of the recessive allele, and recessive alleles are the ones that get masked by the effects of the dominant allele. It's a very interconnected definition. So for example, in the Punnett square that's given below, there's the um, the dominant allele is shown by the capital letter B, and the recessive allele is shown by the small letter b. Now, the capital letter being the dominant means that the brown is the dominant gene, and the blue eyes are the recessive, not the gene, sorry, allele. Brown is the dominant allele, and blue eyes is the recessive allele, which means that based on, I mean, the cross is different, but regardless, there would be a 50% chance of having a child with brown eyes because the brown dominant allele will mask the blue allele, even if the blue allele is there. And then The next is the 3-to-1 ratio. Now, this is the standard ratio that he talked about. Mendel basically crossed two varieties of pea together and found that all of the offspring, or the F1 generation, had the same characteristic as one of the parents. He allowed the F1 generation to self-fertilize, which means that they had the same exact um, pair of alleles. And each plant produced offspring by fertilizing its female gametes with its own male gametes, and the offspring, or the F2 generation, contained both of the original parental types in a 3-to-1 ratio, instead of having only the one of the parents. Now, before we talk about how this works in modern terms, we're going to talk about heterozygous and homozygous. So heterozygous is when you have one dominant and one recessive allele, and homozygous is when you have the same allele. You can have homozygous dominant or homozygous recessive. Homozygous dominant means you have obviously both of them are dominant alleles, and homozygous recessive is when you have both recessive alleles. So in this case, that's a heterozygous allele pair, and that's a homozygous recessive allele pair. Now, we talk about the 3-to-1 ratio in modern terms. This I drew based on, we talked about this last year, no, in the May session, when we were talking about this, someone wanted a visual representation. It's not the best drawing, but it works out. It's two Punnett squares, which show how it works. So, each pea plant has two alleles of the gene that affects the character. So in this case, we're talking about the color. Each pea plant has two alleles, either green or yellow. Now, based on the tests that Mendel did, we figured out that the parents are homozygous because they have two of the same allele. Now, the original parents have to have, one of them had to be a green plant, one of them had to be a yellow plant. For it to be yellow, it had to be homozygous recessive, but for it to be green, it could have been either heterozygous or homozygous dominant. Based on the cross, we figured out that it's homozygous dominant. So, based on this cross, you can see F1 is all heterozygous children. Obviously, since we're crossing one homozygous dominant and one homozygous recessive together, you're going to result always in 100% heterozygous, 100% dominant allele showing or phenotype result. So in the F1 generation, there was 100% a result of green plants, but based on the second test or the self-fertilization test, we can go back and figure out that they were all heterozygous children. Now, based on that, if we go further down, we can see that the heterozygous alleles crossing against each other will result in 3 green plants and 1 yellow plant. And this is because there's one that will be a completely dominant allele pair, two that will be a dominant recessive heterozygous pair, which will still result in a green plant, and then one that will be a yellow plant because it will be a homozygous recessive, which means that's wrong actually. It's not 3-to-4 heterozygous. I'm gonna fix this again. Sorry guys. It's a 1-to-4 homozygous dominant, a 2-to-4 heterozygous, and then a 1-to-4 homozygous recessive. Okay, that makes sense. Now, we're going to talk about Punnett squares. We already talked about that one. I'm gonna make you do a question. There's only one person here. Two people actually. I'm not, and Sin, if you want to answer this, you can. But Leaf, if you want to go ahead and try to answer this question and draw a Punnett square for it, then you can go ahead and do that, and then we can talk about it. But if you don't feel like doing it, you can also let me know, and we don't have to do it. Okay, go ahead. I will wait until you are done. Yep, you got it right. So I'm gonna assume you know how it works, but for people who are watching the recording, it basically works the same way that this works, where when you're crossing two heterozygous pairs, it's going to result in a 3-4 dominant and 1 recessive phenotypic ratio. So it's going to be 3-to-1 phenotypic ratio, but a 1-to-2-to-1 genotypic ratio. I assume everyone knows what genotype and phenotype means. Anyway, now we're going to talk about autosomal diseases. Let me check where we are at in terms of, okay, yeah, we should be fine. Okay, so autosomal diseases. The two examples of autosomal diseases that we need to know is cystic fibrosis and Huntington's disease. Now, cystic fibrosis is usually happens in, when it happens, usually neither parent will have the disease, but they'll both be carriers of the recessive allele for the disease because it's an autosomal recessive disease. So the normal allele is the dominant C, capital C, and the cystic fibrosis allele is a small c. Usually what happens is that when a child is born with cystic fibrosis, then both the parents are carriers, and that means that they will essentially pass down. There's a 1 in 4 chance that they will the cystic pass down cystic fibrosis to their children. There's a 2 in 4 chance that the children will be carriers as well, and there's a 1 in 4 chance that they will be completely normal. Because again, this is a double heterozygous cross. So obviously in cystic fibrosis, since it's a recessive autosomal disease, usually neither of the parents will have it, which makes it really difficult to predict unless they've done some kind of gene sequencing where you know that you are both the parents are carriers, in which case then you will know that you have a chance of having a child with cystic fibrosis. The other disease that we need to know is Huntington's disease. This is a neurodegenerative disease, which is caused by the dominant alleles of the gene coding for Huntingtin, which is a protein with an unknown function. We don't know what it does, but it not being present causes neurodegenerative issues. Now, in this case, the picture I used a double letter to show how it works, but you can just use a capital H and a small h. So usually, since this is a disease that is a autosomal dominant disease, usually the normal is that you will have two normal alleles which are both recessive. However, if one of the parents has Huntington's disease, um, then they will 100% pass it down to their, not 100%, sorry, they will have a higher chance of passing it down to their child. Okay, now we're going to talk about pedigree charts. Pedigree charts are another way, which they basically work like another uh, genetic diagram, another genetic model, um, to basically track disease down a family line. So in this picture, we're talking about cystic fibrosis. Both the parents don't have it. However, they had two children. One of them had it, one of them didn't have it. And then, but then the child that didn't have it had to be a carrier because they passed it on to their child. However, the child that had it did not pass it down to their child. But however, we know that since cystic fibrosis is an autosomal recessive disease, the two children definitely have to be carriers because um, the father will be passing on one of the alleles regardless. Is that because there's no affected parents because of the low? Is that you? No, it's not that there is no affected parents because of the low life expectancy. It's because um it's very difficult. It would be rare that two people who have cystic fibrosis would have children. Like I don't we usually I That's actually a really good question. I don't think it could be about the life expectancy, but we usually, when we talk about um, cases where uh, the child gets cystic fibrosis, it's usually that both of the parents have the heterozygous allele pair. But that's actually a good thought. It might be because of the low life expectancy. Yeah, no worries. Okay. Yeah, we were talking about pedigree charts. So yeah, again, since we know that the parent has cystic fibrosis, he will definitely pass it down to his children. However, he married a normal or unaffected female, and neither of their children had cystic fibrosis, which means that they will both be carriers. If um, this gentleman with cystic fibrosis had married a woman with cystic fibrosis as well, then they would have resulted in children who will 100% have cystic fibrosis. Okay, this is a sample question from a example. I think the answer says it there, but the answer is there. But basically, the logic behind this sample question is that again, okay, so the way I would tackle this question is look at what disease we're talking about. So we know we're talking about cystic fibrosis, which we know is an autosomal recessive disorder. So we know that anyone who's affected will have to have the double recessive or the homozygous recessive pair. So we know for sure, 100%, that the boy has a um, like has a history of not a history, sorry, has cystic fibrosis and therefore has a homozygous recessive pair. Now, when we track back, we know that for the boy to have a homozygous recessive pair, both of the parents will have to have a at least one recessive allele. Yes, which means that this is not a possibility because the if the father had a double A and was completely unaffected and also not a carrier, then none of the children would have had it and would there would 100% they would be either carriers or just completely fine. So we know that because the um, boy is affected, the father is not affected, and this is not a sex linked disease, it has to be C, A, capital A and small a. One of the tricks of this question is that they've specifically asked you about the father because they want to hint at it being sex linked, possibly, because they're talking about a boy and then talking about the father. But we know that it's, again, like I said, the first thing you need to do is look at what the disease is and know whether it is autosomal, whether it's sex linked, or whether it's recessive or dominant. Those are things you just need to know. The recessive or dominant part, you can kind of guess based on the pedigree chart because when it's a dominant disease, if a parent has it, it's very likely that the um, children will all have it. So it becomes kind of difficult to like look at pedigree charts that show dominant diseases. I mean, it becomes really easy to, I mean, what I meant was it becomes difficult to mess up charts that show dominant diseases because oftentimes, since it's a dominant disease, it will pass on to all the children. And then it just becomes everyone is affected. So the first thing, look at the disease, um, figure it out whether it's autosomal or sex linked, figure out whether it's dominant or recessive, and then look at who the question is about. So in this case, it's about the affected boy and his father. But to figure out what alleles the father has, you also have to look at the mom, and then you look at the family that is concerned. Sometimes you might also need to look at the other siblings because they can be carriers or they could be affected, but the boy could not. The question could be about one of the unaffected kids, but then they're talking about the affected people. Another trick, I'm not going to call it a trick, but it's just something to trip you up a little bit, is they don't say from the fourth generation. They say affected boy. You need to be able to read the key. You need to know that the squares are the males, the circles are the females. I need to know that affected male, when they're asking affected boy's father, they're talking about this one. It can't be this one, it can't be this one. So just things to keep in mind, not to trip over when you're in a stressful exam situation. Okay, now we're going to talk about codominance. So, some genes have pairs of alleles where both have an effect when they're present together. So, it's not like a normal pair of allele where because one is dominant, it will automatically mask the recessive. This is situations where the pairs of alleles both have an effect when they're present together. So they're both dominant. These are called, like the definition suggests, codominant alleles. An example for this is the flower color of Mirabilis jalapa, which is the scientific name. But in this plant, if a red flowered plant is crossed with a white flowered plant, the offspring that they have will have pink flowers because the red and white alleles are codominant. So if you look at the notation, pay close attention to the notation. It doesn't work the same way as normal Punnett square notation or normal allele notation that we do. It works as a superscript. So for red flowers, the allele is CR, and for white flowers, it's CW. When you see something like this, it's usually um, either codominance or sex linked. However, if it's sex linked, then it's going to be with an X. It's not going to be any other letter in the normal case. So, codominance, in this case, CR/CW, if they're mixed, then they're going to have pink flowers, which are CR/CW, and they're both codominant. And that's why they're noted like this. Dominance is when an allele codes for an active and functional protein, and a recessive allele is when it codes for a non-functional protein, which is why um, dominance works over a recessive. Because when an active and functional protein is there, then you're not going to be able to see the non-functional protein when the functional protein is active. The main thing we know about codominance is the inheritance of blood groups. Now, this is in a lot of questions, a lot of times, especially in the MCQ. You'll also a lot of times get pedigree charts that are mixed with the inheritance of blood groups. So I'd say the blood group inheritance thing is fairly important. As we know, there are 4 blood groups in the ABO system. There's group A, group B, group AB, and group O. The inheritance of ABO blood groups follows both codominance and multiple alleles. So instead of having just 2 alleles, just black or white, or one of two, there's multiple alleles in the inheritance system. So the alleles of blood groups in the ABO system are as follows. There's 2 dominant, there's 1 recessive. Is the IA, IB, and then the small I. The IA obviously codes for group A, IB codes for group B, but then the small I, or the recessive I, codes for group O. Now, this is a question for Leaf. And, oh, someone new has right. Okay. This is your question for the day. Can you determine the possible genotypes for blood types A, B, AB, and O? So write down what the genotypes are in the chat, please. Are you guys going to try the question? If not, I can move on, but I would like you to try it. You have to write all the possible genotypes of A, B, AB, and O. Yeah, those are the possible phenotypes. What are the possible genotypes? What are the gene combinations or allele combinations that are possible for those blood groups? I-I-O. Okay, so the only issue with that leaf is that there is no I-O, it's small I. So you would never write it in, the I will not be in caps, and it won't be I-O, it will be lowercase I. Yeah, and in A it will be I-A-I, and in B it will be I-B-I, but it'll be small I at the end. So there won't be any I-Os, it'll be I, small I. So that's really important. You can talk about group A as IA, you can talk about group B as IB, the alleles for group A and the allele for group B. However, the group O will always be small I. It will not be IO. Okay, so there's codominance and there's multiple alleles. So in codominance, um, codominance is what results in group AB forming. So the parent phenotype is group A crossing with group B, which means an IA crossing with an IB. This is the situation where we're going to talk about codominance. This is what will result in a group AB. So the IA and IB will result in gametes that have IA and IB. They will not have gametes that are other than those two specifically, since it's completely homozygous dominant parents. Those two gametes will fuse and form IAIB, which is the phenotype, the genotype for the phenotype of group AB. That's where codominance comes in. Multiple alleles in our blood groups is when there is two heterozygous parents. So when we have a heterozygous group A and a heterozygous group B parent, and they have IAI and IBI, lowercase I, they will have gametes with IA or gametes with I, and group B will have gametes with IB or gametes with I, and these coming into a cross into the Punnett square will result in either group AB, group B, group A, or group O. So this is a situation where there's 4 different phenotypes and 4 different genotypes all present, and each combination of the gametes will result in a different outcome. Okay, predicted versus actual outcomes. So this is also an important thing to talk about, because you can predict that 3 out of 4 offsprings will have this. However, in actuality, in real life, the outcome will not be the same in that sense. So, for example, parents of blood groups O and AB have genotypes lowercase ii and IAIB. So the offspring could be IAI or IBI with a 1-to-1 group A to B ratio. So theoretically speaking, or if we predict that if a parent has 2 children, when we predict it, we'll say that one of the children will be group A, one of the children will be group B. However, when in actuality the parents have 2 children, both of those children could have group A and both of those children could have group B. It doesn't have to be that each one of them has one of the groups, because predicted ratios are only expected when the number of offspring is large. So if that specific set of parents, a parent with an O blood group and a parent with an AB blood group, were having children, if they had 100 children, then we could say that 50 of them are going to be A and 50 of them are going to be B. And then even then, there'll be a discrepancy between the numbers. That could be 45 to 55 or whatever. However, when you only have 2 children, it doesn't have to be that it's going to be 1-to-1. The predicted ratios don't always translate to actual outcomes. Now we're going to talk about sex linkage. This often ends up tripping a lot of people up in questions in both and in paper one and in paper two. And it's really important to know how sex linkage works because um, it's fairly easy once you start understanding how it works. So, sex linkage is the association of a characteristic with the sex of the individual. So, sex linkage is when the characteristic, or usually we talk about sex linked diseases. So, when the disease allele is linked to um, the sex chromosome or the 23rd pair. Sex linkage is almost always due to genes on the X chromosome. So in males, there is only one X chromosome, and the occurrence of these conditions that are sex linked, are a lot more frequent in males since they only have one chromosome, one X chromosome. So since they're sex linked, if a um, parent, if a mom is a carrier of a disease and the dad does not have the disease, um, their children, if, for example, they have three children and they're all boys, their children have a higher probability of getting the disease because it basically becomes a 50-50 chance because they get the Y chromosome from their dad and they have a 50% chance of getting the diseased or the sex linked disease allele rather than the normal sex allele of X, which is why males are usually um, affected and females are end up being the carriers rather than the um, affected. So in humans, hemophilia and red-green color blindness are the two main examples that we learn about that are conditions due to recessive alleles of sex-linked diseases. Now, this is a question for you guys who are here still. We're gonna do a pedigree problem which talks about sex linkage. So here we're talking about hemophilia, and the pedigree chart below shows the inheritance of hemophilia in a family. I want you guys to try all three of the questions that are there. Do you guys want to try it? I'm gonna wait like 2 more minutes. If not, I'll keep going. Okay, let's do this then. So, the first one, person one, they are over here. And the way we will tackle this is based on their children because they're from the outside of this family. We know that the mother is a carrier, but we don't know anything about their history. Okay, so we know that the mother is a carrier, so we know that dad has, the child has a 50-50 chance of getting hemophilia. However, um, we don't know what the case is with the dad. So, um, to figure that out, we look at the daughter. The daughter is not a carrier based on this key. She is a normal female. For her to be a normal female, she needs to have the X capital H, X capital H pair. For her to have both of those means that she got the good one from her mom, and that means that the dad also has the normal um, X chromosome. So that would mean that the dad was unaffected, an unaffected male. It would be a normal male. Does that make sense? Leaf. Okay. So it wouldn't be X small h y because she's the carrier, but she's not a carrier, which means that she had to get both of the good dominant um, alleles. Okay. Question two. Determine the genotype of the mother of person two. Now, we look at them and we have to determine their mothers. So, based on the fact that uh, sorry. For number two, we have to look at their siblings. We can't just look at number two because when it's numbered, it doesn't necessarily follow the key here. So we know that the daughter is a carrier, which means that she had to get the X lowercase h gene from somewhere. We also see that the child is the hemophiliac male child. Sorry, the child is hemophiliac, so that means that he has the X lowercase h y gene, which means that his father gave him the Y, and his mom definitely had the X lowercase h allele because she had to have passed it down to um, the child. However, to determine whether or not she was 100% affected or a carrier, we look at the number two. They're a normal male, which means that they had to have gotten a normal X chromosome allele from somewhere. So if they're a normal male, if one of the males is hemophiliac and the one female in the family is a carrier, it would mean that the mother was also a carrier, which, Leaf, your answer is correct. So the mother would have an X capital H, X small h, sex chromosome pair. For number three, we look at the third person. So if person three has a son and the father is a hemophiliac male, predict the son's phenotype. Now, to determine the phenotype, first we have to determine the genotype. To look at the genotype, we know that the father is a hemophiliac male. In order for him to be a hemophiliac male, he has to have the X lowercase h y um, chromosome pair, right? Which means that he has the X lowercase h that will pass down, that will be there, and the Y. However, the son will have a will get the Y chromosome from the father, which means he will not get the hemophilia gene from the father. Now, here, to determine whether or not 3 is a carrier, because we don't always know whether the numbered persons follow the key. We know that neither the mom nor the dad are a carrier or a hemophiliac male, which means that they have to be clean. There's no way for the child to have uh, uh, the female to be a carrier, because neither the mom nor the dad were carrier or hemophiliac to begin with. Now, since she's a normal female and the dad is a hemophiliac male, the hemophiliac male will only transfer down the Y chromosome, and the normal female will pass down the X capital H chromosome, which means that, and she has both of those, which means that no matter what the genetic variation, she will always pass down the normal chromosome. So the son's phenotype will be non-hemophiliac, he will not have hemophilia. Okay, now we're going to talk about causes of mutations, and then we talk about DNA technologies, and then we're done. Okay, so causes of mutations. There's many things that can cause a mutation, but the main one is radiation and mutagenic chemicals. So these will increase the mutation rate and can also cause genetic disease and cancer. Radiation increases the mutation rate if it has enough energy to cause chemical changes in DNA. So there are certain rays from radiation that will not cause mutations, but there are certain that will cause mutations. So gamma rays and alpha particles from radioactive isotopes, shortwave ultraviolet radiation, and X-rays. These four are 100% mutagenic. So these have enough energy to cause chemical changes in the DNA. However, there are certain rays that do not have enough energy that will cause chemical changes in your DNA. So it can't cause mutations. Now, some chemical substances can cause chemical changes in the DNA and therefore are mutagenic. And these chemical substances, when you are exposed to, can therefore cause cancers and genetic diseases. An example is benzoprene pyrene, which and nitrosamines, which are found in tobacco smoke and mustard gas. So tobacco smoke, obviously we know that smoking eventually leads to cancer. So it basically is because of these two chemicals which can cause chemical changes in our DNA, cause mutations, and therefore cause an extreme division of your cells, which will therefore cause cancer. However, these were also in mustard gas, which was used as a chemical weapon in the First World War. So after the First World War, there was a large population that turned out to have genetic diseases and cancer. Okay, last bit, DNA technologies. That's, I think, yeah, we're literally just going to talk about this and then we're done. So we have 5 main DNA technologies that we talk about in our syllabus. It's gel electrophoresis, PCR, DNA profiling, genetic modification, and clones and cloning. So the first one is gel electrophoresis. Gel electrophoresis is basically a method of separating mixtures of proteins or fragments of DNA which have been charged. So the mixture of our DNA, the DNA sample, is placed on a thin sheet of gel by attaching electrodes to both, which acts as a sieve essentially. And then an electric field is applied to the gel by attaching electrodes, as you can see here, and here, to both ends, depending on whether the uh, particles are positively or negatively charged, they will move towards one of the electrodes or the other. So basically, the way it works is there's a tank, there's some fluid in there, you put in two electrodes, there's some gel, and you place the DNA sample in a well in the gel. And then once you do that, because of being charged positively or negatively, they will move towards one of the electrodes or the other. Usually, DNA is the more negatively charged the DNA molecules are, they will move further towards the positive electrode. So, based on um, their size, the rate of movement will change, and therefore there will be a track seen. So, certain molecules will go further because um, of their size. So the smaller ones will move a lot faster than the larger ones through the gel, and therefore um, be further close to here than there. The larger bits are here, and therefore you can separate the fragments of DNA by size. The next one is polymerase chain reaction, or PCR. So PCR consists of a cycle of changes carried out again and again to produce many copies of a DNA molecule and expand it so much so that you can see it and figure out the patterns. So PCR consists of a cycle of stages carried out again. I already read that out. There's three main stages. Firstly, the DNA is heated to 95 degrees Celsius to separate the two strands. So we basically do the work of, this is me recalling the transcription helicase, I think. It's basically what separates the two strands. And then when it is separated, the temperature is then reduced to 53 degrees C, which allows primers to bind to both strands of the DNA next to the sequence that is to be copied. Once these primers have been bound, the temperature is increased back to 73 degrees Celsius, which encourages Taq DNA polymerase, which is a type of DNA polymerase that is captured from a bacteria that live in spring water, which means that they're in hot springs, which means that they are a lot, their optimal temperature is a lot higher, which is why we move it up to 73 degrees C, which encourages them to replicate both strands, starting at the primer, producing two double-stranded copies of the original DNA. Now this is repeated again and again. I don't know if you can. Probably should be able to. Anyway, this is repeated again and again and again until we get multiple strands of the DNA stacked together, which it'll be millions of copies. And this can all be done within a few hours. And this becomes very useful when very small quantities are sampled and a large amount is needed for analysis. So take the DNA, you do this, multiply it essentially, and then you can take small samples and do multiple testings rather than when you don't have the choice of going and collecting more DNA by any chance. Okay. The next is DNA profiling. In the DNA, there are short tandem repeats, otherwise also known as STR, which are loci with short sequences of bases that are repeated many times. So a sample of DNA is, the way DNA profiling works is a sample of DNA is obtained from a person. It must not be contaminated with DNA from anyone else or any other organism. So you would have to take it with gloves, and it has to be put into a secure vial. And yeah, so the DNA from a selection of STR loci is copied by PCR. The DNA from between 11 and 13 loci is copied in commonly used DNA profiling methods. So it's very unlikely for two individuals to have the same number of repeats at 11 and 13, and therefore by using the DNA from these loci, you can determine whether a person is, who the person is. The copies of STR alleles made by PCR from one's DNA is separated by gel electrophoresis. So DNA profiling is basically where gel electrophoresis and PCR are both used together. And the result will be a pattern of bands. So something like, I don't know if I put in a picture, but it's a common question because we get questions with different profiles, and then you have to determine who the parent was or who the culprit was based on how common their profiles are. So two individuals are extremely unlikely to have the same exact pattern of bands unless they're identical twins, but they're between parents and children, they'll have similar pattern of bands, which means you can determine whether someone is their parent or not. Okay, genetic modification or GMOs. Genetic modification is the transfer of genes from one species to another to create genetically modified organisms or GMOs, or otherwise also known as transgenic organism. Trans being mixed or changed, and genic meaning genes. Genes are transferred between species using a vector, which can be plasmids, like we talked about earlier from the eukaryotes. So the plasmids are used as vectors, which means the gene is inserted into the plasmid and then cut out of the plasmid. So there's two main enzymes that we use in this process. There's restriction endonuclease and DNA ligase. Restriction endonuclease is what cuts the DNA molecules at the specific base sequences so that the gene can be attached. And then DNA ligase makes the sugar phosphate bonds that links the nucleotides together and forms the continuous strands of nucleotides and basically links them all together. A plasmid with a gene from another species is inserted. A plasmid with a gene from another species inserted is called a recombinant plasmid. The cell that receives the gene is a host cell. So any plasmid that's used as a vector is also known as a recombinant plasmid, and the cell that receives the gene is the host cell. The risks and benefits of GMO crops is a fairly common question that you can get. It can be a 4-mark question in a paper too. Usually I'd say it's either a 4-mark or a 3 or 4-mark, I'd say probably, in a paper too. So a gene from bacterium has been transferred to some varieties which codes for a bacterial protein called Bt toxin. This kills insect pests feeding on the crop, especially corn borers that can cause serious damage. However, this very gene has its own benefits and risks from being transferred into GMOs. So, the benefits. There will be higher crop yields and thus more food for humans because there will be less pest damage. The pests will be killed just by feeding on the crops. There would also be less land needed for crop production. Some of it, since there will be less pest damage, so then you can lessen how much area is being used. Once that happens, then you can use some of the areas for wildlife conservation. There is also the chance of less use of insecticide sprays, which are expensive and can be harmful to farm workers and to wildlife that is naturally occurring there. And instead of using insecticide, you can let the insects feed on the crop, but they die anyway, so then it basically prevents the damage and also prevents having to use insecticide. Some of the risks are that insects that are not pests could also be killed. There could be native insects that are actually beneficial for the environment that are killed, and then that would cause a disbalance. The transfer gene might spread to populations of wild plants by cross pollination, which would also make them toxic to insects feeding on them, which is not great because we don't need the insects to all die. It's just to avoid the crop from being ruined. And then the third one is the insect pests of corn may develop resistance. Resistance is a very real thing. It once resistance is formed, then it becomes basically useless to have that bacterial protein in the plant. One of the other risks that you can talk about is that it could possibly be harmful to humans in the long run, but it's not the best risk to put in there, but it's one of the risks that people talk about often. Okay, the last one is clones and cloning. So a group of genetically identical organisms derived from a single original parent cell is a clone, and production of an organism that is genetically identical to another organism is called cloning. Asexual reproduction is a natural form of cloning. So many plant species, some animal species, prokaryotes can do this. They just do mitosis. They produce genetically identical cells to create whole new cells of their own, and they become basically any single cellular organism does cloning usually, but some animal species can also do it. Many plant species can also do it. Breaking up an embryo into more than one group of cells at an early stage would mean that each group of cells develops into a separate genetically identical individual. At the embryo stage, the characteristics of an animal are mostly unknown. So you could take an embryo, break it up into more than one group, and then that would mean that they are genetically identical, but they will develop into completely separate individuals. But the characteristics are mostly unknown, which means that you could be cloning an embryo that would have gone on to die. So one of the ways of cloning that we have attempted is Dolly the sheep. It's somatic cell nuclear transfer. So what happens in somatic cell nuclear transfer is that a cell taken from the udder of a donor adult is taken and cultured in a lab for 6 days. An unfertilized egg is taken from another sheep, the nucleus is removed, and the egg cytoplasm and the structure is left behind. The egg without a nucleus is fused with the donor cell using a pulse of electricity, which means that the donor cell's features, organelles, and most importantly, nucleus will be fused into the egg cell. And that means that instead of the egg cell having a haploid nucleus, we've now given it a diploid nucleus, which means that it basically becomes a zygote of its own. When it becomes a zygote of its own, it can then become an embryo as it divides and becomes its own organism, essentially. But the embryo that results from that will be transferred to a third sheep, which becomes a surrogate mother, and then that surrogate mother will give birth to a lamb. Now that lamb is called Dolly, and she is genetically identical with this sheep. Not this one that donated the egg cell, not this one that was the mother, but this one is which donated the nucleus, because the nucleus is the part that determines the genes of the sheep. And that's it. That's the end of our entire genetics topic. Do you have any questions at all? If not, then I'm happy to end this here. That's basically everything you need to know about genetics. If there are any specific questions for people who are watching this after, if there's any specific questions, feel free to ping me in the biology chat, because I will then be able to look at it. If you don't ping me, I'm not always on it, so I can't see it. But if you ping me, I will look at it. I will look at any questions you have. And it doesn't have to be about genetics. It can be about anything. And I will be happy to answer it. Okay. I'm going to stop recording now. And then